Welcome everyone to our CARTI Seaweed series. Today, I'm joining everyone from the traditional territories of the Wewekai, Wewekam, and Quirka nations, where we're fortunate to live, work, and play. I encourage everyone to just acknowledge quickly where they are and the territories that they're all on as well. We are recording this meeting, so it will be available online on our projects page on the CARTI North Island College website. So if you'd like to share this around to anyone and everyone that you think would be interested after, or people that have been unable to join today, please feel free. And a big thank you to NSERC as well for being able to help us put this project on and, share some cool information to a bunch of different people about seaweed. It's been really fun so far, and we've got some cool speakers today. Joining us from the East Coast, we have Scott Lindell from Woods Hole Oceanographic Institute, and we have David Waring, who is joining us from Cornell in New York. So they have some pretty cool presentations ready for us today. In addition to having a listen and eating your lunch, if you're doing that, I think I saw Christina snacking on some kelp snacks. Feel free to maybe drop your location where you're joining us from into our chat box on the side there. It's kind of cool to see the different locations that everyone's coming and joining us from. Otherwise, I don't think I have too much else to say other than feel free to hang out, eat your lunch, and just listen in to our presenters today. But we're going to start off with Scott. He's going to talk to all of us about our kelp, or about his kelp breeding program and some work that he's done on that. And then we'll move into David's presentation. And then we will open the floor to questions for both of them. So I think their presentations are going to be about 10 or 15 minutes each. So yeah, I'm going to hand it straight over to Scott now. And yeah, just hang out and have a good time. Thanks, guys.
Can you hear me now?
I can hear you.
Okay.
Absolutely, I can.
And can you see my full screen?
I can, yeah.
Okay. Let's test this. Does it go forward and back?
I see your 4-year summary page.
Right. Trying to get to the next one. How do I do that?
There are the little arrow keys down the bottom if you need to use that on the bottom left of your page there. Maybe that'll be an easy way for you to do it.
All right. That's what I'm going to do. Well, Thank you for joining us over your lunch hour there on the West Coast. I grew up on the West Coast, so I'm still getting used to the East Coast time, even though I've been living here for, I don't know, half my life now. So I'm going to give you a quick summary, so we have time for questions and conversation too, of the last 4 years of selective breeding with sugar kelp. This was a program funded by the Department of Air and Energy's Mariner program, where they're laser focused on potential feedstocks for biofuels at some point in the future, but knowing full well that there'll have to be some intermediate markets, and those intermediate markets right now are mostly food and perhaps animal feed someday, and maybe bioplastics. But let me go into sort of the general project objectives and a little bit of introduction to the cycle of selective breeding. So we collect wild sporophytes initially to sort of develop our founder population, and we'll take some time to isolate individual gametophytes, that's males and females, and I'll describe how that's done momentarily. And then we want to exercise whole genome sequencing on those gametophytes when we get enough biomass to begin to detect variations in the sequence. And then we'll make these crosses between males and females, plant them out, and we'll do a phenotypic evaluation of those sporophytes. That is what we call characterizing the variation and the performance of these, the genetic background of these sporophytes, which we feed into a statistical model, which then we can use for genomic prediction. And then we start this whole virtuous cycle of selection and, oh, this thing is going automatically. That's very upsetting. Then we start this whole cycle of selection, characterization, selection, and selective breeding to field test and improve the next generation. So here's a sort of a nice pictogram of the selective breeding cycle in a form you can probably better understand it. Again, this thing is still going automatically. So we will. It's very annoying. We'll go out in the initial founding population. We'll go out into the wild, collect this blade that you see on the far left, excise the source tissue, release spores into sterile seawater in a beaker. And we've developed automated ways of cell sorting. And this sort of starts my list of what are the key advantages of working with gametophytes in selective breeding of kelp. So we can use automated flow cytometry, cell sorting technologies to put those into 96 well plates. So we have separate males and females growing in those plates. Those males and females can be expanded into flasks. So we have these uniclonal cultures of both the males and females. And eventually we'll have enough gametophytes that we can have enough biomass to create a cross that's meaningful. For us, a meaningful cross is something that fills about 1 meter of grow line. And that whole cycle of growth takes about 6 months before the cycle starts again in the laboratory. But these other advantages of gametophytes are that they can be maintained for decades or cryopreserved. We can maintain them at low light and red light, low light and low temperature. And we can build up a virtual unlimited supply of clonal germplasm, particularly with new technology that's being developed right now, these photobioreactors, because we can keep them in that vegetative growth growing long term. And the most important and sometimes critical things for farmers and hatcheries is that we have independence from when seed or source tissue normally ripens in the environment. So it seems that with warming oceans now, it's coming later and later that you can go out and collect ripe source tissue in the fall. So being independent of that and be able to start your hatchery 3, 4, maybe even 6 weeks earlier and plant earlier could be key to extending the life, the growing season and the yields. So we've come a long way in just 4 years, but this is really just a blip on the evolution of seaweed breeding and farming compared to modern agriculture. So we've collected this very diverse germplasm now, and we've made it publicly available at the Bigelow Labs here in Maine. So everything we're doing is pretty much open source. And we've got now, based on some population genetic studies we've done, we actually have 2 regional breeding programs, one north of Cape Cod, where we're situated, and one south and west of Cape Cod, what we call southern New England. So, and we actually have 2 different research farms for that purposes. So we don't cross that line of where the biogeographical regions of these different strains lies. And we've characterized this germplasm and all our field trials by, putting them into a publicly available database, which David Waring will talk about after I'm done. We call it SugarKelpBase, and you can go to that and see all the gene sequences and all the different growth trials we've tried. We've got something like 50 different traits that we're measuring for each plot. Everything from blade morphometrics, length and width and thickness, to plot level traits like biomass weight, number of blades per cross or per meter, and then about dozens of traits, or sort of composition traits of the blades as well. A suite of sugars and minerals and obviously carbon and nitrogen as well. I think one of the things we're most proud of from this project is being able to find a way to keep these selectively bred strains as they evolve into farther and farther from something that we've been used to grow or harvesting in the wild, to keep them separate from the wild populations by creating seedless varieties as well. And I think this will be important for meeting the regulatory requirements of preserving wild populations, from which we depend on this genetic diversity to create improved strains from farm populations. Sorry, going back one more time here. And I fully want to recognize some of the challenges, one of them being to educate and bring regulators along for understanding what constitutes a risk in terms of using local strains versus what kind of improvements should be allowed before a population has to become seedless in order to be planted. Also note that the hatchery process using gametophytes still requires some fiddly work. We often have to contend with asynchronous fertility between males and females, and the application of the gametophytes to ropes or strings sometimes results in uneven densities that we'd like to be able to control better. So a little deeper dive into some of our accomplishments. As I say, we started with no a priori knowledge of which crosses to make. We did just basically crossed many things at random to begin to build this big data set that we can begin to sift and sieve to find these correlations between different genomic traits, genomics, and the traits that we see on the farm. So our population genetic study consisted of 18 locations, and you'll see I've listed the publications that will give you a deeper dive into any of these topics. They're at the end of this, the full citations are at the end of this talk. We've bred and field planted different families or crosses in common gardens, both in New Hampshire and Connecticut. So about 1,000 crosses thus far over the last 4 seasons. And then we've presented the phenotypic and genetic analyses of the first 3 of those 4 years in 2 other publications. And with our partners at Cornell, we've developed and applied a genomic selection tool, which gives us much higher precision and accuracy in selecting for traits such as yield, percent dry weight. We hope to apply it to sugar content soon as well. And we're beginning to select strains for their temperature tolerance, which I think will be really important. We've developed a reference genome too, which we hope to soon publish, and will be a great reference for others following in our footsteps to conduct their own selective breeding programs for their regions. So we've got about 18 different crosses thus far that have produced fresh yields that are over 15 kilograms per meter. And I'd say that's at least 2 times the industry average. Our chief king plot here was 28 kilograms per meter fresh weight. That's about 4 kilograms per meter dry weight. And this is a summary of all 4 years. The first 2 years being random crosses, the last 2 years being more directed crosses. And you can see at the the far end of this bell curve, the 18 crosses that represent those that were over 15 kilograms per meter. One other key finding was that we have actually two different types or morphologies, and more recently the skinny kelp has actually been deemed a separate species of sugar kelp. But when you cross the skinny kelp, this very skinny morphology, with a typical sugar kelp type morphology, we're getting more than 20% average yields higher, showing some hybrid vigor. So coming back to the topic of, can farm strains impact the wild? And obviously they can, but at what point? Right now, the relative abundance of farming to wild kelp is quite small, and at the size of plots that we're using for our quote-unquote selective breeding program, they're just a meter long, and they're all from locally collected strains, at least geographically similar strains in the Gulf of Maine or southern New England. It's unlikely that you're going to have much of a risk of interbreeding at this scale. But we have to start planning for what we might see are, cornfields like cornfields, farm kelp in the ocean. If it does grow to be a bigger and bigger enterprise, it could, in local areas, dwarf the population of wild kelp. And so we've elected to use our bioinformatics and whole genome sequencing to screen for and create populations or interbreed strains that have natural mutations that confer basically sporophytes that can't produce fertile gametophytes. And we've proven this to be successful. It took many crosses to actually get just a few to work, but we now have at least 6 different strains that are putatively sterile or infertile, and those are planted again this year to confirm we get the same results when we harvest in June. So the real key thing about these seedless strains is they unlock the regulatory and commercial pathways for large-scale kelp farming. This is not to say that you need seedless kelp for the size and scale of the industry right now. I think we'll still see small, local, regional farms near shore providing beautiful product, and seedless strains may not be necessary for them. But if we're going to even move offshore and produce hectares of kelp farms and use selectively bred strains that could double, triple, quadruple the yields, then we're probably going to need to have commercially tailored, improved crops. We're going to need to address these environmental issues, and seedless will probably be an important trait. And we know that just looking at our progress over the last 4 years versus the 50 years of more traditional breeding that has been conducted in Asia, we're moving at a markedly faster rate than they ever did. And so we've patented, or we've applied for a provisional patent for this process of selecting sterile or seedless kelp, more as a way to prevent a Monsanto from grabbing this territory. It's one that we want to make more freely available. So these are the 6 crosses that we harvested last June, and we didn't see any visible source tissue on them, so we held representative blades in a special induction tank where we kept the water relatively cool, the day length short, and we were able, over a course of 4 to 6 weeks, verify whether these strains would actually develop source tissue and whether that source tissue would actually produce viable gametophytes. And these 6 did not produce viable gametophytes. I will note that one of the crosses was one of our highest yielding crosses in 2021, as well as being one of the higher yielding crosses in this 2022 harvest. So it might be nice if we actually have the confluence of both high performance and seedless kelp in at least one strain. So as I say, we've been trying to make this open source and make sure that we can capture the advances going forward here. Our innovation for this seedless kelp was awarded a gold innovation at the Aquaculture US conference last September. And this was the announcement that our collection is now available at Bigelow Labs for others to select from. Our current field sites range from New Hampshire to Virginia at this point. We're going to be very busy harvesting from these 5 different farms over the course of the next few weeks. And here are the list of publications. I know that Logan will make this recording available afterwards. And I want to recognize the project team representing folks from Hui and UConn, USDA Cornell, our partner farmers who we couldn't do this without at Greenwave and at UNH, University of New Hampshire, and our engineers at Cliff Gowdy and Associates. And one other shout out, well, also we got to acknowledge our funding agencies, Department of Energy, Mariner Program. We're also continuing to get support from the World Wildlife Fund. And one other shout out is that we're from, knowing you guys are from British Columbia, many of you, these modular seaweed bioreactors I really think are the wave of the future. And we've been growing our gametophytes in some of these bioreactors for about a year now, very successfully. And I'm saying that not just because I got this wonderful T-shirt from Industrial Plankton, but they are doing some great work with for the seaweed hatchery industry to come. And with that, I'll conclude.
That's amazing. Thank you, Scott. I love your T-shirt.
One more time.
Cool. I think that we'll swap straight over to David. And as you mentioned in your chat, we'll get a little more information about the kelp bank, sugar kelp bank. And then we'll open the floor for questions just afterwards. So I'm gonna just highlight David here and spotlight. And I think if there we go. There we go. Okay. Hopefully everyone can see the presentation and we can all see David up here or over here maybe. So yeah, go for it, David.
Great, thank you. Yeah, so my role in the Mariner breeding program that Scott was presenting about is to serve as a data curator and software developer for SugarKelpBase. And what I'm going to talk about today is give a sort of very high level overview of what SugarKelpBase is, some of the different data types that can be stored in this database, and then a simple example of how to use the data in the database to kind of find some interesting information from it. So at its core, SugarKelpBase has these 3 main components. The first is a database. So this allows us to store all the data that's collected by the breeding program. And this database is on a server, so it's not on an individual computer. It is on a server, so it is available to anyone that needs to access the data. The second component is a website. So this is how most people access the data in the database. This website can be used to view the data. You can upload new data and manage and modify any existing data within the database all on this website. And then the last component is an API. And this is a standardized way for third-party tools, different analyses tools, or data download tools to access the database through this standardized way. And SugarKelpBase is publicly available, so all the data is publicly available on the database, and you can find it at sugarkelpbase.org. Now, we didn't create SugarKelpBase on its own specifically for kelp breeding. We're using a software called Breedbase, and Breedbase bills itself as a comprehensive breeding management and analysis software. And it is developed by the Lucas Mueller Lab at the Boyce Thompson Institute, located at Cornell University. And it is open source and freely available. So if you're interested in it, you can go to GitHub right now and download all the source code and run it yourself if you're interested in doing so. For more information on Breedbase itself, you can go to breedbase.org or read this paper that was published last summer in G3 by Nick Morales. And it goes into all the details about how Breedbase is developed and the different ways of storing its data. Some benefits of using Breedbase. So there are other breeding databases available, but Breedbase has some really strong benefits. One of those is that it has a very strong track record. It's been around for over 10 years. I think it's close to 15 years at this point. It's still being actively developed, so there is a core team of developers that are hired just to work on this software. And there are other breeding programs that are making contributions to it as well. So because it is open source, there is a community of developers working on it. So other breeding programs that are actively using the database will create new features and make improvements and make those contributions available back to Breedbase so that way other breeding programs can benefit from it as well. It's currently used by many crops. I have a few listed right here. So the Next Gen Cassava breeding program is a large user of Breedbase. So they have a version of Breedbase called CassavaBase, where they store their cassava breeding program data. It's used widely by yams, and then also by a U.S. Department of Agriculture project, which I work on, called the Triticeae Toolbox, and we store wheat, barley, and oat data in that. And we're getting more and more interest in other breeding programs using Breedbase as well. So you can see from these crops here, it was originally designed for terrestrial field crops, but it is sort of flexible enough where you can use it very easily for kelp and for other species as well. So there's a USDA breeding program in salmon that is interested in using Breedbase too. So it is very flexible in the types of data and the types of species that you can store the data in for. There are a lot of benefits of keeping all of your breeding data in a database. The main one there is that it can easily be shared with collaborators. So instead of having one person sort of maintain an Excel spreadsheet with your data and then passing that on through email and worrying about whether or not you have the latest version of that spreadsheet, the latest version of all the data is always available through the website that you can access from anywhere. All the data is stored in a uniform way, so that makes it very easy to compare data across years or locations. And it's very easy to combine or subset data from different sources all within the database. And data is also accessible to additional third-party tools. And again, that's through that API, which uses a standard called BRAPI, which is the Breeders Application Programming Interface. So this allows tools and different databases to communicate with each other using a standardized protocol. And it makes it very easy to get data in and out of the database using these other tools as well. Here's a list of some of the primary data types that are stored in Breedbase or in SugarKelpBase, and I'll go through some of these in a little more detail. So the first data type are locations. So we have GPS coordinates for all of our trials and field sites in the database. So you get a nice map of all of your locations. All of your trials are directly linked to a location in the database. So you can pull up a specific location and then very easily find all the trials for that location. We have specific data types for accessions or germplasm. So some of the metadata that are stored for accessions include synonyms. So you can keep a list of any sort of alternate spellings or alternate names for specific germplasm. We can link to external databases through these registered accession numbers. You can add notes and comments, and then it also stores pedigree information. So for a specific progeny or a specific cross, you can link that cross directly to its parents, and then you can use this interactive pedigree viewer to navigate through the pedigree tree. So you can click on these purple arrows here and see, all the siblings or all the parents and navigate your way through the pedigree tree right on the website. For trials, we have some basic metadata such as the location of the trial, the planting and harvest dates, any notes or comments on the trial. And then we're also able to save the trial design and layout as well. So we have the design type information such as the blocks and the reps, the controls that are used. So in this example here, these purple plots are the controls. We can store the field layout, so these are the positions of each plot relative to each other. And for kelp, we have one row equals one line that's put out in the water, and then each column is a position along that line with a specific germplasm in each of those plots. And those plots are linked to accession entries in the database. So you can pull up a plot and very easily pull up any information that you need about those specific accessions in the database. For the trials, we can store the trait observations. So the trait observations can be made on different levels. So you can have plot level information, such as, the overall plot wet weight. You can have individual blade information, such as the length and width measurements. And you can also store subplot level information. And the table here, you're seeing the summary stats for a single trial. So it's very easy to see your means and ranges and standard deviations for all the traits that were observed in that trial. And then you can also get a nice histogram to see the distribution of all your trait values. So in this example here, we're looking at individual sporophyte wet weights, and you can see the distribution of those values. And on the website, if you hover over a specific value, you can get the accession name here. So you can easily pull out some outliers from that. You can see which one has the really high wet weight here. Another data type that's used quite a bit in the SugarKelpBase are seed lots. So originally, seed lots were created to keep track of the inventory and the physical seed packets that people are storing in their labs. But for SugarKelpBase, we're using seed lots to keep track of information on the maintenance of gametophytes that are kept in the lab. So here we can keep track of individual water changes for containers of gametophytes, as well as information about their condition. And we have an easy way to record that information. So SugarKelpBase can generate barcodes for any information that's stored in the database. And then this seed lot maintenance tool can scan those barcodes using an integrated barcode reader. So you can pull up all the information for that associated seed lot. And then you can quickly record any of the actions or observations that you take on that seed lot just by clicking these buttons here through this sort of simple UI. So it's a quick way to record any sort of maintenance that you're taking on gametophytes in the lab. So once you have all this data stored in the database, there's a lot of tools available for downloading the data or doing simple analyses. So in this example here, we're using the selection index tool. This is a very simple selection index. Here we've selected a single trial. We're interested in a single trait here, so I've selected the plot wet weight. So we can very easily find the accessions that have the highest wet weight in this trial. And if you're adding multiple traits, you can add coefficients to those traits too, so that way you can weight the traits differently. In this example here, we found this S69 cross has the highest wet weight in that trial. And you can click on the link of this accession name here, and you can pull up more information about that individual accession that had that highest plot wet weight. And here we can get a table. So this is on the detail page for this one specific accession. And here we have all of the traits that have been collected for that accession and its average value and the number of times those traits were observed. So it's a nice way to get an idea of how that specific accession is doing. And then if you're interested in its pedigree, on that same page further down, you can look at the pedigree of that accession. You can find the parents that were used in that cross. And then if you click on these purple arrows here, you can find its siblings as well. And then if you're interested in downloading data, it's very easy to do that using the search wizard tool on the database. So this allows users to combine data of different data types and sources into custom data sets. And then those selections can be shared and used later, or you can download that data directly from the search wizard. So this is what the search wizard looks like. You can filter data with either one to four different criteria, starting with the left column here and moving to the right. In this example here, I've selected accessions as the first filtering criteria, and then I've selected those 3 top performing accessions from that selection index we did before. And then we can see what traits have been observed for those 3 accessions. In this case, I've just selected 3 traits that I'm interested in. And then you can use this related trial phenotypes section down at the bottom here and download data for those 3 accessions and those 3 traits all on this page here. And that download is in a CSV file. So it's a very easy way to get data out of the database using the search wizard here, and a nice way to be able to combine data from different trials or different accessions and locations and subset it to just the traits that you're interested in. So that is all that I have. It's, again, just a very basic and brief overview of what SugarKelpBase can do. If you are interested in learning more about it, feel free to contact me. I have my email address here. You can also go to the website directly at sugarkelpbase.org. And then if you're interested in Breedbase in general, you can check them out at breedbase.org or contact Lucas Mueller here. And with that, I'll stop sharing and hopefully we'll have some time to answer any questions.
That's awesome. Thank you, David. There's so much information in those Breedbases. It's super cool. It's such a valuable resource for when you're doing things like this. Yeah, super cool. Do you also have a I Heart Comedified shirt or something similar?
I don't. I am very jealous, though.
You need an I Heart Breedbase shirt.
Yeah.
Yeah, absolutely. There are a couple of questions in the chat. If anyone else has any questions, there's a little like raise your hand section at the top up here. So just click that and then we'll get through to you to answer some questions you guys might have for either of our 2 presenters today. But the questions that we have in the chat, let's get to those. I have one from Mike Spranger, who I think is in Washington, looking at his previous message. Mike asks, can you anticipate when and if work will be done in such a way that West Coast growers and hatcheries can benefit? I see Nina has posted a link for Alta Seeds Conservancy, who I think that Scott, you might be a little involved with.
Sure, let me have a quick endorsement here. Yeah, the Alta Seeds Conservancy has sprung from a sister project on the West Coast with giant kelp, but it's not focused entirely just on giant kelp. They'll be looking at a variety of West Coast species from Alaska to California. I don't think they're ignoring BC. I hope they will include you guys as well, of course.
Yeah, up here.
Yeah, and so yeah, that would be a good first contact for some of these genomic tools, especially. I will say that we have a project in Alaska, and we're intending to collect more sugar kelp varieties from Alaska as well, and we'll be sharing those with the Alta Seeds Conservancy. So all this work developing the tools that Mariner program has generated are going to be spread nationwide and hopefully internationally too. We can't get enough kelp, I think, for all the low carbon replacements for rather high carbon lifestyle.
Yeah, that's awesome. That's really cool that you're getting lots of different varieties into the project. So I really appreciate that. Chris Song, who I think is in Alberta, has a question for you, David. Are there plans to include nutritional data in the Kelp seed bank?
Yeah, and Scott, you can correct me, but I believe we already have some of that in the database, right? We have some nutritional analyses.
Yeah, we certainly have. We have a full suite of sugars for the breakdown of the carbohydrates. We have carbon and nitrogen. We have metals. So we don't have arsenic for every plot. It's quite expensive to analyze, or iodine for that matter. But we do have other quote unquote heavy metals. But we don't. I mean, I guess this wouldn't be probably good enough to put on your your packaging. It's not what we're measuring isn't the traditional way you would put on a consumer package, let's say, but it would give you certainly a very good indication of the likely nutritional quality of any of these crosses.
Cool.
That should be a good proxy for protein, for instance. Yeah.
Awesome. Leanne, I think, has her hand up and has a question.
Okay. Hi, David. Hi, Scott. Thank you so much.
Hi.
Totally enjoying your presentations. The last presentation on the software trials, blocks, reps, controls. So your software illustrated it in little blocks, literally. But when
Yes.
your trials are out in the field, are they on long lines? And how long is the average standard trial size?
Yeah. So typically we're using what we call a 5-line array. So we have 5 200-foot long lines spaced about 3 feet apart. So it's a pretty tight area that we're farming. Does that answer your question?
Sure. May I add to that?
Sure.
How do we, if different farm trials are happening in different locations and each farm has a different design, how do we compare those?
Well, I would hope you would adopt ours. I'd hope you adopt ours because it's quite elegantly simple. It only requires 2 anchors and a 10-foot spreader bar on either side, which they're not very expensive. And Cliff Gowdy, who I referenced, provides those. There's dozens of those in Alaska now on farms. And the key, though, that David pointed out is you do need to block it in such a way and have reference crosses that are spread through those blocks. So we'll take one reference cross and make 10 of those plots, 1 meter plots, throughout the 200 to 400 plots that are in the farm.
Logan, may I ask one more, please?
Yeah, go for it. No,
Okay.
it's question
Regarding
time.
direct seeding, are you direct seeding your spools and then transferring them onto the grow line, or are you direct seeding your grow line? And also, is the binder seed ratios, have you had some success there? I know you have, that you could speak to. Thank you.
For our breeding program purposes, again, we're dealing with what we call pucks. They're these tiny, relatively tiny spools, maybe 4 inches tall, wrapped with string, and we literally paint them with induced gametophytes, juvenile sporophytes, and then we roll them in a binder, the ATSI binder that's been used in Europe for many years, and then we still incubate them for about 4 weeks before they go out into the field.
Thank you for that.
Yeah, that's how we ensure that we get good coverage before we put things out.
I happened to do a similar experiment and cured it on the grow line, planted it out, and had various success depending on the ratio. But one particular ratio worked really well.
Interesting. We'll have to compare notes because we've done other direct seeding trials directly onto rope and out into the field, and some things have worked and some things haven't, and I don't understand why sometimes. So maybe we can piece it out.
I
another
really
time.
appreciate chatting with you today. Thanks.
Cool. If there are any other questions, just pop your hand up in the chat. Scott, I don't know if you have a minute or there, but you've been using the gametophyte bioreactor from Industrial Plankton, who are actually on the island here. I don't know if you have a minute to talk about how much you've enjoyed using that little machine.
Yeah. Well, this was all very new for both us and for Industrial Plankton. We got the first set of 10-liter, what we considered commercial scale size photobioreactors, and we quickly filled. We have 3 of them. We quickly fired one up with an assortment of gametophytes to bring the density up as quickly as we could. And yeah, within 3 or 4 months, we went from, I think, 1 or 2 grams inoculation to 100 grams. And I figure that 100 grams would, I think if this is numbered right, yeah, would seed about 10 kilometers of rope. So it's very compact. What they are generating now, I think quite cleverly, is they've developed a more research based model. There are 2-liter modules, which would probably suffice for the average farm nowadays too. But they package them in 4 packs basically. So an associate of mine in Maine is going to be working with an 8-pack system with some of our strains likely. And I think it's going to be a wonderful tool to facilitate this strain development on a more less than the 1 meter scale that we're working at, and hopefully the 10 and 100 meter scale, where we can see real progress in development.
Nice. So you see it as like a really useful tool for both the farming aspect as well as the research aspect.
Absolutely. The nice thing about these photobioreactors is you can control light and temperature and most importantly, carbon.
Okay.
By manipulating the pH, you can control the amount of carbon that are easily accessible to the gametophytes, which is a key thing that they need besides nitrogen to grow quickly.
That's awesome. Yeah. No, I had some interest in it just because they're super cool machines, and they're also like pretty local to us in BC. So yeah, they're just so cool.
Yeah, they've obviously learned a lot in their microalgae world and been able to pivot and turn it into a macroalgal bioreactor.
Absolutely. No, it's a fantastic piece of machinery. Cool. I don't see any more hands up, and I think we've gone through all of the questions in our chat here. So I think we might start wrapping things up there. Thank you again for everyone joining us, and especially thanks to David and Scott for your presentations and sharing with us. It's really cool, both what you guys are doing, and especially here out in the BC coast, where we're starting to get the kelp world up and running. It's really taken off here, so it's super applicable stuff.
I don't know how many of your...
Oh, I think you might have just got muted there, Scott. Sorry.
I was going to say, maybe you've already know this, or maybe your audience already knows this, but the next International Seaweed Symposium will be in Victoria in about 2 years. That will be a good opportunity for me to meet many of you if I haven't already.
Yeah, absolutely. Yeah, that's going to be a really cool event. I think we're all looking forward to it on the island here, and yeah, it's a great time to come out here and explore. So if you're coming out this way, you guys let us know. Actually, anyone in in the meeting. Let us know, and let's hang out and talk about some kelp things. And I'm pretty sure there are some pretty cool kelp snacks already available on the island, and I think at that time there might be even more. So maybe we'll just have like a kelp sampler, or like, put a movie on and eat some kelp snacks. But yeah, I think we'll wrap it up there. Thank you again for everyone for joining. And again, yeah, it's going to be available online. So check out the CARTI projects page. I'll send an email out probably either this afternoon or tomorrow, just reminding everyone where that link is. We do have a meeting next month as well. I think that the Aquaculture Canada conference is on around the same week, so I'm just trying to fiddle with dates and times to find a good spot to slip next month's presentation in. But keep an eye out for an email about that, and yeah, share the link far and wide for anyone that wants to see the previous recordings, this one, and any future events. So yeah, thanks everyone for coming by, and thanks again, David and Scott.